Hi everyone, my name is Josh and I'm going to tell you about how I'm taking an engineering approach to turbocharging proteins. Proteins are like cars. Most are made out of a standard set of parts and they work just fine. But some proteins are turbocharged to perform special tasks. So, come with me and I'll tell you about how I'm going to engineer proteins. Cells produce thousands of different proteins in order to carry out their most essential functions. Most normal proteins are composed of 20 standard parts called amino acids, but some extra important proteins are turbocharged with a special 21st amino acid called selenocysteine. Any protein that incorporates this special building block is referred to as a selenoprotein, and they often have enhanced properties like improved speed, stability, and durability. These enhanced properties are incredibly valuable. For example, humans produce 25 selenoproteins, and defects are associated with a wide variety of health concerns. Additionally, researchers want to harness the power of selenoproteins to create new synthetic ones with applications in things like producing renewable hydrogen energy, improving crop hardiness, and helping us understand how we can engineer cells. But it's actually quite difficult to produce selenoproteins in the lab because they follow a uniquely complicated biosynthesis pathway. This dramatically limits both disease and synthetic biology research since we can't even make the proteins for study. So for my thesis, I'm excited to create a technology that allows for robust selenoprotein production, similar to improving the assembly line that then produces race cars. This is what the selenoprotein producing factory looks like at a biological level. Importantly, a molecule called a transfer RNA, or tRNA, is at the heart of every single step. I'm going to make targeted changes to the tRNA in my first approach, similar to upgrading specific machines in a race car factory in order to boost production. We're going to look at an interaction between the tRNA and a protein called EFTU. We can do this by engineering the tRNA in the specific region that interacts with the protein in order to improve it. We can measure the improvements in our tRNA by measuring how cells fluoresce differently. In this case, the better the tRNA variant, the more green fluorescence there is. I also have a second approach that attacks this problem from a slightly different angle. This would be analogous to giving the factory workers themselves a huge incentive for finding any way they can to improve production. This technique is called directed evolution, and here we engineer a virus so that the only way it can survive is if it's able to make selenoproteins, but it also might not. So over time, the viruses adapt to this pressure by evolving their tRNA sequence, and we can figure out what mutations occur so we can improve selenoprotein production. Combined, these approaches should remove the difficulty in making selenoproteins. Now you know my plan for turbocharging proteins, and we're going to leave everyone else out in the dust.